Good day, everybody. In this series of lectures, we'll be talking about the plasma membrane, also known as the cell membrane, with regards to its structure and its function. Because in biology, we always have to assume there is some, the function of whatever organelle there is related to its function. And in this particular lecture, we're going to be looking at the structure of the cell membrane itself. Now, it's important to remember that cells are trying, just like human beings, are trying to maintain homeostasis. Homeostasis literally means same state because it comes from the Greek and Latin term here, homeo, which means same, and stasis, which means like a state of balance. So you put these things together, homeostasis, the cell is trying to maintain a steady state or a state of balance where everything stays within physiological norms. As an example, your internal body temperature is 98.6. And for the body to work properly, we have to maintain a temperature somewhere close to that. So if we go outside on a really hot August day and the temperature is too hot and our body starts to heat up, we will begin sweating to try to cool off the internal temperature. So sweating returns us to our homeostatic temperature of 98.6. Now the cell membrane, as again is this outer covering here, all around the outside, has three distinct functions that we're going to talk about in this chapter. The first one is support for the cytoplasm. Now, by this I mean that the cell membrane is responsible for allowing some materials to pass through, either in or out. That way we say the cell membrane is selectively permeable. So it supports the internal environment by allowing some things in and some things out. The cell membrane is also responsible for recognizing foreign materials. As we'll talk later on today, the cell membrane has proteins in it, and these proteins, some of them have glycolytic properties, where you're going to have some particles of glycogen attached, or little sugar molecules attached. That makes the cell a little different between people. So a person with type A blood has a different glycoprotein than a person who has type B. And the cells can recognize this. Our immune system can recognize self from non-self and attack anything that's foreign. Third thing cells do, the cell membrane functions for, is for communication. Cells communicate by sending signals to target cells. One of these signals could be when the cells of a tissue are pushed up against the edge of a petri dish, for example. They send signals to stop growing. The same thing would happen if you had a cut in your skin. As the cells regenerated, the cells don't continue to regenerate after the wound has been closed, but once the wound is closed and there's no more space for the cells to fit, they send signals to stop growing. They stop dividing, and hence you don't have the cells continue to grow even after the wound is healed. In order to really understand how the cell does all this stuff, we have to look closely at its structure. The cell membrane is not solid. It's a lipid bilayer, which means this. When you look at it, the cell membrane is composed of two layers, an outer layer and an inner layer. And each layer of these phospholipids has a head portion, which is polar, which means it's going to be attracted to water, and a nonpolar tail, which means it's going to be repelled by water. So in this case, with a lipid bilayer, you have a layer of polar molecules, portions of the molecule that are polar, each one facing the water-loving side, and the nonpolar tails all in the middle. Now the terms we use here, hydrophilic and hydrophobic, remember the prefix hydro means water in each case, but philic refers to loving. So heads are basically water loving, while the tails, these portions down here, are hydrophobic or water fearing. This particular arrangement allows only small nonpolar molecules to pass through. Now the next slide is going to show a little video that kind of sums things up to this point.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions such as sodium ions and hydrogen ions cannot pass through the membrane unaided.
So the important thing to remember from this, if you saw that video, was that if the molecule is, large, in order for it to pass through, it must be lipid soluble. If it's a large but polar, it won't go through. Now we're going to take a look at how those other molecules can get through, despite being insoluble through that phospholipid bilayer. This is accomplished by the different membrane proteins that are present in the membranes, and they float inside this phospholipid bilayer kind of like boats on an ocean. Now, unlike boats on the ocean, some of these are going to face only the inside of the cell, such as this one down here. Some are going to face only the outside of the cell, like this one right here. Some are going to stretch in between the two, like this one right here. And these would probably be the really critical ones here, because these are going to be the ones that are going to form little channels that will allow the ions and the large polar molecules to slide through. And some of them are going to have carbohydrates attached, like these that I've shown over here. Again, these are the markers here that are going to actually identify, certain types of cells. Those would be the ones that would identify, for example, the blood types I mentioned earlier. To help you organize your thoughts on these protein types, I've compiled this little concept map here to help you visualize what's going on here. If you take all membrane proteins, they can be divided into 4 distinct groups. Those that move materials across the bilayer are basically going to be called the transport proteins. And they will be either tunnels or have specific shapes that will allow molecules and basically ions to pass through. Some membrane proteins are going to label cells. We call those cell surface markers. Others that monitor the environment are basically the receptor proteins, while those that facilitate diffusion are different forms of enzymes. This summarizes the way that proteins can assist getting particles through and control the communication and flow of information between the different cells.